Thank you. So to kick it off, I'm just going to share some statistics for you about what's happening in Virginia right now with research and development, specifically that which is focused on healthcare. So interestingly enough, we are number 15 in terms of federal funding. That's mostly made up of funding from the National Science Foundation, the CDC, and the National Institute of Health. We also have quite a bit of private sector investment in this area. We had over $300 million in venture capital that was invested in research and development projects related to healthcare over a 4-year period. There are also really great examples. For example, back in December, there was a $50 million gift to Inova for cancer research. In addition, we have a great pipeline that's being developed. In a 4-year period, we had over 1,400 patents that were secured around the biosciences. Another really interesting statistic is that all the research that takes place in Virginia universities is related to healthcare. So that's great. Very active. And here's just a slide about the investment from a federal funding standpoint. So what this means is that this research and development sector, specifically in healthcare, has a tremendous impact on the Virginia economy already. The total employment in these establishments is more than 26,000 individuals that are directly employed for those establishments, and another 100,000 that are also related to these establishments. The average wage of $77,000 is significantly above the average wage for Virginians at $51,000. So there's no question about the positive impact that this sector has on the economy of Virginia. But I think what you're going to find today is there's even another impact that's more important, and that is the impact that these developments have on creating, saving, and improving lives. So with that, what I'd like to do is turn it over to John.
Okay, so I'm a professor at UVA in cell biology, urology, biomedical engineering, and obstetrics and gynecology. And since I'm basically a lab geek, my talk today is going to focus on basic science discovery and the ways in which identifying biomarkers in the sperm and the egg are leading to first-in-class new products that are impacting the Virginia bioeconomy. So my title is Putting the Signature Molecules of the Sperm and the Egg to Work. So what you may ask is a signature molecule of the sperm or the egg. So this red worm-like structure that you see up there with 2 eyes is actually human chromosome 11. The 2 eyes represent the location of a gene called ACRV1 on each of the 2 sister chromatids that make up chromosome 11. This ACRV1 gene was discovered and named at UVA by researchers in my lab. The ACRV1 gene encodes a protein called sperm protein 10, or SP10 for short, and SP10 is located in the sperm head. If you look at that apple green fluorescence on the anterior part of the sperm head, that shows you the location of the SP10 protein. Now this is a view inside the human testis, where the sperm are made. And the testis is organized into structures called tubules. They're called seminiferous tubules, and there's 1 seminiferous tubule there displayed on the screen. The black stain localizes the position of this SP10 protein. Now, what you see is that the SP10 protein is restricted to just 1 cell population right against the lumen of the organ, and that population are the spermatids. The cells that differentiate into sperm are underneath them, and they're not stained at all. This particular protein is expressed at a very specific stage of sperm maturation as the spermatids mature into the mature sperm. So this is another sort of geeky slide. It shows you along the top all the organs in the body. You can see testis over there on the left, and walk down it, there's kidney and lung, etc. So what we did was to take RNA from all the organs in the body and probe it for the expression of the SP10 protein and gene. And if you look over on the left, you see the bands that indicate the presence of the ACRV1 gene expression. But as you look under all the other tissues, this protein is restricted in its expression to the testis, and not another organ in the body is making this protein. So there were lots of other pieces of evidence, along with the ones that I've shown you, that were taken to the FDA to qualify this particular biomarker as a testis-specific protein. So we went down the development path, and we created monoclonal antibodies to the SP10 protein. You see two of them there, and they were used to bind to the SP10 protein and to begin to measure it. And there was a direct relationship between the presence of the SP10 protein and sperm concentration. And then we moved into that immunodiagnostic cassette that you see on the right, striped the monoclonal antibodies into those cassettes, and we then began to quantitate sperm numbers with a test very much like the home pregnancy test. So this led to the founding of a Charlottesville biotech company. It's now doing business as Sperm Check, and it led to the first FDA products to use in the home to immunodiagnose sperm. And the Sperm Check fertility test that you see here takes about 10 minutes to perform. It informs men whether they have normal or low levels of sperm, and it directs them to a fertility specialist if their count is subfertile. The Sperm Check test addresses the needs of couples to identify the underlying causes of their infertility. The test allows male factor infertility to be identified in the privacy of the home. And as most of you know, about 40% of the time when a couple is experiencing infertility, the cause does lie with the man, and 90% of the time that's usually associated with low sperm count. So Sperm Check Fertility is stocked on the family planning shelves at Walgreens, CVS, Rite Aid, and Target. It's now in Boots pharmacies across the UK and Europe. It's sold in 17 countries, and this UVA-originated discovery is becoming a global brand. The product is identifying male factor infertility earlier in the process when couples seek to pinpoint the underlying causes of infertility. And as you might imagine, this device is emerging as a women's healthcare product. Women, not surprisingly, are buying the product and taking it home for what we might call the reluctant male. So right now, more than 2,500 kits are being sold every week somewhere in the world, and Sperm Check is just one example how identifying these signature molecules in the sperm are being put to work. And other products that are going to be emerging out of the discovery of sperm and testis-specific proteins include their use as male and female contraceptives and their use in forensic science. Now I want to shift gears and talk about some applications of egg biomarkers. This is a cross-section of an ovary, and it localizes a protein we call SAS1B. The protein is localized with a brown dye. So can everybody see that you got these round structures staining in the ovary and nothing else is staining? This demonstrates really something quite marvelous. This particular protein is restricted just to the eggs. And if we zoom in onto the surface of the ovary, you're going to see something even more amazing. The brown stain on the left reveals that the SAS1B protein is first found in eggs that have reached what's called the secondary follicle stage of growth. The small dormant eggs, which you see on the right, the primordial eggs, the ones that have not yet begun to grow, the quiescent pool of eggs in the ovary is negative. These are the reserve eggs that cycle during every recruitment phase in the ovary. The dormant eggs that you see there are not making the SAS1B protein and are not staining brown. So among all the normal organs in the body, this protein is only made in the pool of eggs that have begun to develop and grow and then to ovulate. Now, if you look in the picture on the right, the lower image, it shows you an ovulated egg. And if you look above it, you're going to see a green stocking cap-like domain on the egg surface. That is the domain that this protein lies in at the membrane of the egg. Now on the left are uterine cancer cells. And one of the things that we've discovered is that a large number of cancer cells begin to express proteins that are normally restricted to the egg. It's a new insight into the fundamental nature of cancer. Aside from cancers, as you see here, this protein on the surface of cancer cells, this protein is normally restricted in the body just to the egg population. So what does this mean for cancer therapy? It offers us a powerful new paradigm for cancer treatment. Because the SAS1B protein is found on the surface of tumor cells, but among the normal organs is restricted only to the pool of growing eggs, this provides us a way to deliver drugs selectively into tumor cells while not harming other normal, healthy cells in the body. And since this protein is absent in the pool of dormant eggs, treatments that target this SAS1B protein are predicted to spare the reserve of quiescent eggs and allow fertility to return after the treatment ends. So a new Charlottesville spin-out biotech company has been formed to capitalize on this discovery, and this discovery is now leading to innovative new therapies. We're making hybrid drugs that consist of two parts: a biological component of a monoclonal antibody, as you see there in red, that selectively binds to this target on the surface of tumor cells, lethal payloads consisting of DNA intercalating drugs linked by a linker arm to the monoclonal antibody. And this strategy, of course, is referred to as antibody-drug conjugates. We're also making imaging agents labeling these monoclonal antibodies with radionuclides. So how does this work? The antibody is infused into the bloodstream of the patient, and then the antibody binds to this SAS1B protein at the surface of the cancer cells. The drug payload is released, and this results in tumor cell death. So this type of antibody drug directed to a tumor surface protein represents, of course, a form of targeted therapy. And to conclude, I'll just give you a view of the numbers of cancers that we have found and the various incidences that this new target is present in. It includes some of the most insidious and hard to treat and retractable cancers like ovarian cancer and pancreatic cancer. It's estimated that more than 340,000 patients every year will be impacted by this discovery, and it represents a very significant economic opportunity, a multi-billion-dollar market. So I hope that this brief glimpse into some of the signature molecules in the sperm and the egg has demonstrated how basic research on the genes that are being expressed selectively in the ovary and the testis and finding their way into the sperm and the egg is leading to both new intellectual properties, new spin-out ventures, and breakthrough products that are impacting the healthcare landscape in Virginia and beyond.
Well, good afternoon. I want to thank Barry and the Chamber for hosting this event. I tell you, when you hear about research like what's being done at UVA, you're going to hear about at Tech and Inova and other parts of the state. It's just so impressive. I mean, the ecosystem here is great. I moved back from the West Coast after 25 years, and I am blown away at some of the great healthcare work that's being done here. So it's a real blessing to be back, and it's real exciting to be here. What I want to do is talk a little bit about our company, Kaleo. We feel very, very fortunate to be building our company here. When we got started, everybody told us we had to move to California to raise the money. And we said, no, we want to do it in our hometown. And they said, well, you're not going to be able to raise the money. You're not going to be able to find the talent. You're not going to be able to Well, we're making some pretty good progress. We got a lot of work to do, but we're really pleased to be doing it right here in the Commonwealth. So most life science companies are started by scientists, by investors, by clinicians. Our company was founded by patients. It was founded by Eric and Evan Edwards, who grew up with severe life-threatening allergies to all seafoods, dairy products, peanuts, tree nuts, some antibiotics. They were labeled the most allergic children that their doctor had ever taken care of. And you probably know somebody or have personal experience with food allergies or bee sting. Raise your hand if you've had some experience with life-threatening allergies. Yeah, I mean, it's rare that we're with a group that a very large percentage. And so, as you know, people with these allergies live in fear that they'll get served something that they're allergic to in a restaurant, or they'll get stung by a bee, and then all of a sudden they're in a fatal situation within minutes. And so these patients have to carry an epinephrine auto-injector with them all the time. The problem is they're bulky, they can be embarrassing to carry, and they can be difficult to use. And so these two young guys in high school had the vision of, couldn't we build a credit card sized product that would provide epinephrine when somebody's life depends on it? And that was the impetus of the company. And so we, since that time, have built a company that has 2 FDA approved products. The first one is what their vision is. So this product, AviQ, is the size of a credit card. It's about the thickness of an iPhone. And the whole purpose is to be small enough to be with you wherever you go, to be really simple to use. In fact, it talks to you, it tells you what to do, and then has a needle that delivers the drug but then retracts so the patient never sees the needle before, during, or after. And so I'll demonstrate it for you real quick with this trainer. So when you purchase one of these, and you should all get one of these, by the way. You should all have one of these. When you get one of these, you get 2 of these, and you get one of these, which is a trainer. And the trainer functions exactly the same, but it doesn't have a needle and drug. So I'll demonstrate it for you. So you take it out of the case. First thing it's going to tell me is there's no needle or drug, and then it'll walk through as the actual product does.
This trainer contains no needle or drug. If you are ready to use, pull off red safety guard. If not, to inject, place black end against outer thigh, then press firmly and hold in place for 5 seconds. 5, 4, 3, 2, 1. Injection complete. This trainer may be reused for training purposes.
So it walks the patient step. Now, I do know that's not my thigh. Just wanted to clarify that. Just want to clarify one thing. It walks the patient step by step through the whole process. So we launched this product in January of 2013, and what we're most excited about is that 27 days after we launched it, a high school girl saved her own life with the product. And since then, we've had many reports of that, but it never, ever gets old hearing about particularly a kid that saves a life. So today what I want to do is talk a little bit about Kaleo. I want to talk about our second product, which addresses a huge, huge problem in this country. And then I'll talk a little bit about where we're going in the future. So as we launched this product and got it into the market, we licensed it to a big pharmaceutical company because it took a big commercial footprint to get the product on the market. And there were some capital strategy reasons that we wanted to do that. So they're now launching that product. It's doing extremely well in the US and in Canada. But we started thinking about what are other big challenges we could take on. And what we noticed was that one of the #1 causes of accidental death in this country is drug overdose. In fact, drug overdose has surpassed automobile accidents as the #1 cause of accidental death. And it's primarily driven by opioid overdose, both both by mistake taken prescription opioids as well as illicit opiates. And so we have built a product to address this problem. And in 2013, according to the Centers for Disease Control, over 16,000 people died from prescription opioid overdose. So that comes to about 44 a day. So during the time we're together at our luncheon panel, 2 people will die in this country from prescription opioid overdose. Now, heroin gets a lot of attention, and heroin overdose has increased significantly, but prescription overdose is about 2 to 1. So for every heroin overdose, there are 2 prescription overdoses, and most of them are accidental. Now, in addition to deaths, there are a number of people that have near misses, that have overdoses and end up in the emergency room. And here's where it gets kind of scary. On average, 3,300 children under the age of 5 experience opioid overdose, end up in our emergency rooms across the country. The cost of this obviously is very significant. So 50% of these patients are admitted. Of that percentage, about 10% end up on long-term care, advanced life support. And most of these patients end up being in a hospital about 3.5 days, and it costs about $30,000. So there is a huge mortality issue, but also a huge morbidity and cost issue. And we think it's important to address. Now, beyond the obvious challenge of opioid addiction, there are a number of risk factors that really play in, and we think are the principal drivers to the problem. First is comorbidities. People that have chronic respiratory problems are far more susceptible to have an accidental overdose. Additionally, taking other drugs along with the opiates, so taking Valium, taking a benzodiazepine, predisposes a person to be in much higher risk for accidental opioid overdose. And then if people take high-dosed, extended-release, long-acting opioids, they're at a 2 to 3 times higher risk of accidental overdose. And so there are a number of factors beyond addiction that are important. There are basically 4 patient populations that we think about in the overdose category. The first are people that are taking it for legitimate reasons, and that's the majority of people. So 100 million people in this country have chronic pain. It's the number one reason people see physicians. So they're taking opioids for moderate to severe pain. The problem is some people do become dependent and can become addicted, and so they may get fired by their physician who doesn't want to contribute to that problem. And so then those people often end up in the street trying to buy prescription opioids, and then if they can't get them or they're too expensive, they end up on heroin. So there are 4 patient populations here, but unfortunately some patients migrate across all 3, all 4. Now, but the key thing is that as a medical community, we need to be cognizant of the benefits of opioids, but also what is some of the safety factors and safety things that we need to think about. The biggest risk to taking opioids is that the opioids block the signals of pain to the brain, but they also can slow down the respiratory system. And they can slow it down and they can actually stop it. So when someone overdoses, they actually suffocate. They stop breathing. Hypoxia sets in, as you all well know, in about 4 minutes. Unfortunately, EMS takes about 9.4 minutes in this country on average in the cities. So early intervention is critically important. Fortunately, there is a drug that reverses opioid overdose. It's called Naloxone. It's been around for 40 years, and it's been available in hospitals. Most of you know about that in your health centers. It's also been on ambulances. The problem is that's not where people overdose. People overdose at home. And most of them overdose in front of a friend or a family member. So what we've done is we've built the first FDA-approved Naloxone Auto-Injector that reverses opioid overdose, and very importantly, is approved for use by family members, by non-medically trained people. So when someone accidentally overdoses at home, they can get the intervention quickly, and then they call EMS, and then they get to the hospital. And the difference in outcome is profound if someone gets Naloxone on board within minutes versus 9, 10. So it's extremely important. So we have brought this product to market. It was approved last year. It's our second FDA-approved product. It's an opioid antagonist indicated for emergency use of known and suspected opioid overdose. We're rolling out across the country right now. And then as we look forward as a company, we're really focused on a number of different products in the pipeline. We do keep our pipeline products confidential. We've had to go through some patent litigation, those kind of things. I did mention earlier, we have 165 patents in our portfolio that support and protect our patent portfolio. But in life sciences, you have to have strong patents, and so that's an important part of what we do, and we keep things confidential. But we're very excited about where we're going in the future. One of our fundamental beliefs is that the patient is truly the expert. And so we bring the patient into the development of all of our products, and we're excited about the future. And it's a real blessing to be here today. It's a blessing to be in the state of Virginia building a company, and thanks very much for your time.
Hi, everyone. Is my talk up? All right, so I'm Rob Gordy. I'm from Virginia Tech, and specifically the Virginia Tech Kirlian Research Institute. And if you're curious about how I sound, yes, I'm from the South. New Zealand, as it turns out. So this is an overview of the Virginia Tech Kirlian Research Institute in Roanoke, Virginia. And it's associated with the new Virginia Tech Kirlian School of Medicine there. And our focus is on medical research, particularly medical research that has practical or what we call translational outcomes that may be of benefit to patients. Our executive director is Mike Friedlander, Dr. Michael Friedlander. And if you've ever met Michael, you know that he's a very energetic man. He's also very bright. In fact, so bright that he was named the second most smart person in Roanoke by the Roanoke magazine on Monday. And if you're curious about who the second equal was, it was our local congressional representative, Bob Goodlatte of the 6th Congressional District. So there are three themes at the Virginia Tech Kirlian Research Institute. Neurosciences, which is by far the largest block, the cardiovascular and regenerative medicine theme, which I'm a part of, and also another theme on virology, immunity, and cancer. So I don't have time to introduce every team member, but this is the neurosciences block. We have some very distinguished scientists who have been recruited in over the last 5 years, including experts on addiction, such as Warren Bickel, who runs an addiction center, research center at the Institute, Reed Montague, who is an expert on brain imaging, particularly as it relates to human behavior and economics and markets. Rosalind Moran, who was recruited from University College London, who does computational modeling of cognition or thinking. And the wonderful Craig and Sharon Ramey, who are some of the world's foremost experts on early childhood development. And then our newest recruit, a world expert on brain cancer, Harold Sondheimer, who's from the University of Alabama. They didn't like the fact that they lost him, but he's coming to Virginia, and we're very much looking forward to him being part of our team. So over the last 5 years, according to an NIH metric, the Institute's had about $250 million economic impact on the Roanoke community. And this accounts for sponsorship grants and the various spending that's associated with the running of the Institute. Okay, let's move forward. All right, so I've introduced the Institute. Now I was going to talk a little bit about the story out of my own lab. I originally had a different cartoon up. It was the one about the miracle occurs that was used earlier, so I thought I'd better drop it out and replace it with another one. But it's instructive because, as a basic scientist, I've learned a thing or two over the last decade or so about getting something commercialized. And some of the things I've learned have been good, some of the things I'd have preferred not to learn about, but nonetheless, it's been an interesting journey. So, my lab does basic NIH-funded research, and about a decade ago, we were mainly focused on electrical activity in the heart, and in particular, the type of electrical activity that results in heart attacks. And we were working on a gene that was initially discovered in fruit flies. The fruit fly, as many of you probably know, is very important to biologists. And so we were developing compounds that could target the activity of this gene. And the compound that one of the compounds we developed, we called alpha-CT1. And it's, this thing's got a pointer. It's the wiggly thing on the left. And you can see the fruit fly gene over there, Z01. It binds nicely into that little pocket. I will say, though, that we call it alpha-CT1, but the company that licensed it took it away and market tested the name, and now it's called ACT1. So there's some progress. So the actual practical side of the work started with a very talented scientist trainee, Gautam Gatnikar. So Gautam had done his a veterinary degree in India, then came across to NC State to do his PhD, and then came to my lab for further training. Now, this gene is important in electrical activity, but it's also important in the way that cells move. And again, this is basic research, and Gautam was using a simple culture see you later, Gautam. Using a simple culture method in which we'd grow a layer of cells on a culture dish, scratch them with a pipette tip, and then watch the rate at which the cells came together. And we noticed that this compound was speeding up the rate at which these simple wounds were closing. Now, I'll spare you the details of the animal work in mice that we did, but sure enough, when we did wound healing studies in mice, we noticed a similar effect. The peptide was speeding up the rate at which the wounds were healing. So about this point, Gautam said to me that he really didn't want to become an academic scientist. He wanted to start a company and move into the biotech space. So he made that difficult transition from science to becoming a business professional. I so much enjoy. I stole that cartoon from Tony Atala. He uses it in his talk. I can't claim, but yeah, it is a funny one. But anyway, we started first string research about 10 years ago. And now it's moved beyond just being a biotech startup. It's a clinical stage biotech. We've raised around $20 million in funding from investors, and also around $8 million in grants, including grants from Virginia organizations, including the VBHRC, led by Mike Gresham, and also the CIT, and also some very big grants from the NIH, small business grants. It's been very, very helpful. The company has completed 4 clinical trials, and I'll get to the clinical trials in a second. And just this year, the Food and Drug Administration approved us moving forward to pivotal phase 3 trials. And we've also attracted some top talent to the company, including Professor Bob Langer from the Massachusetts Institute of Technology. So, the company is doing well in terms of its movement forward. So, these 2 phase 2 trials I'm going to focus on are on diabetic foot ulcers and venous leg ulcers. Now, obviously diabetes is a growing problem, and around 10% of folk with diabetes will develop a diabetic foot ulcer. And these are awful, slow-healing wounds, and sometimes they get so bad that about 1 in 10 people with a diabetic foot ulcer will have to have an amputation. Venous leg ulcers are also not much fun. They result from venous insufficiency in the lower leg, and they're particularly problematic and painful in the elderly. So the phase 2s involved around 180 patients. Thankfully, there were no adverse side effects of the study protocol. But importantly, what we found was that Act 1 halved the healing time for both wound types. So the results of the clinical trials were published this year in peer-reviewed journals. This is one of them. This is the one for the venous leg ulcers. And so the company is now getting ready, as I mentioned, to move into phase 3. And I guess the final thought I want to leave you with is that fruit fly research is important because it could help lead to drugs that cure bad and slow-healing wounds, particularly in elders and diabetics. So thanks very much for listening, and that's it.
Good afternoon again. I'm Brian Hayes. I'm the director of the Innova Center for Personalized Health. I want to thank Barry and the Chamber for putting on a very important conference to put together both healthcare as well as the science aspect of all the excellent work we're doing in Virginia. This is the part of the program that if, for those of you like me, had to struggle to get through high school and college biology and chemistry, you can relax now. By the way, I'm just glad I don't have to talk about sperm and eggs, but I do have to talk about business. Let me tell you a little bit about what ANOVA is doing. I assume that many folks understand that ANOVA is a very large regional healthcare system. We have been involved in R&D research for many years, 30 plus years, of a variety of areas, as you can see, with liver and obesity, cancer, cardiology, neurology, et cetera. But our primary focus in the last 4 years has been in genetic medicine or genomics. We decided in 2010 that really one of the key aspects of the future of medicine is genomics, and made a decision to base a lot of our future healthcare delivery on genomic discoveries and the application of that, in other words, translational medicine, into healthcare. So we made this substantial bet since that time. We've put in $125+ million into our Institute for Translational Medicine. We now have 80 scientists working in genetics, and the results have been really very interesting. We've had 6 major programs, one on preterm birth, trying to marry up discovery in the genome with healthcare data, lifestyle data, to predict which particular expectant mothers are likely to have or more likely to have a preterm birth event, which, as we all know, is both a traumatic event in terms of the mother and child as well as in terms of the financial impact. We're now at about 80% predictability, not quite ready to produce this as a product, but for all of us who are concerned about those kinds of things, that's the area of predictability that we would really like to get to. One of the other major studies we have is our childhood longitudinal study. We decided that we really want to be able to discover what is normal, not just what is sick. And the way to do that is you have to actually start with children, actually the fetus in utero, where we collect the genetic samples and we do whole genome sequencing. For those of you who are experienced in that, that is the best you can do in terms of full data. But we also couple that with exome sequencing and a variety of other data that we collect, including marrying it up with our electronic health records and lifestyle data such as weight and various other factors. The result is a massive database of very valuable information. This is mostly from trios. By that I mean it's the child, the mother, and the father. And in some cases we have extended family, including grandparents as well. So this data is allowing us to track events in childhood. We would like to be in this position to track them for 18 years, up to 10,000 children, which is actually 30,000 patients when you think about it, or candidates, so that we can have this massive database about understanding what is normal and be there when something goes wrong. Be able to understand the origins of some of those diseases. So these are 2 of our primary examples. We had an opportunity, though, to take it to the next level. For those of you who are familiar with Northern Virginia, you may or may not know that Exxonmobil had its headquarters in Northern Virginia until 2012, and then they decided that the weather in Houston must be better than the weather in Virginia. Lord knows why else they would go, but they decided to consolidate all of their employees in Houston, which gave us an opportunity to acquire the Exxonmobil Campus. Now, for those of you looking at the slides here, on your left, that's actually north, so obviously on your right, it's south. You can see the beltway along the top. The Exxonmobil campus is 117 acres of land right in the heart of Fairfax County, on the beltway, on Route 50, 1.7 miles from the closest metro stop. It is a fabulous piece of property for anybody, but if you see down in the bottom right corner of your slide, that's Inova Fairfax Hospital. That's our flagship hospital. So it's a great piece of property, but for us, it's a really great piece of property because it's right across the street. So what are we going to do with this property? By the way, it's 1.2 million square feet of office space. I don't know, 26,000 parking places. It's a lot. But what we are not going to do is build a hospital. What we are going to do, or what we are doing right now, is building a center for personalized health. And note the choice of the word health. It's not personalized medicine, it is personalized medicine and personalized wellness. Because we're trying, like many of us in the room, to redefine the entire spectrum, the continuum of what is health. So that we start with our populations and look at what their wellness is, and as we all understand with the change in economics, it's now really pertinent for all of us to keep people out of the hospital. So we're in the unusual position of working very hard to keep people from coming to our place of business. But that's the direction, and that's, we understand, is very important part of what we're trying to do. If you can see on this chart, one of our first major efforts is our Dwight and Martha Char Cancer Center. As Anna mentioned, we just announced a gift of $50 million on May 18th from the Char family, and that is a major step towards building a brand new cancer center. We will be building 74,000 square feet of linear accelerator space because we can't actually take a Exxonmobil office building and renovate it for linear accelerators. But we're going to take an entire office building, about 180,000 square feet, and convert that into our cancer center. Also, one of the major efforts here is rethinking clinical care. We're working very hard on using genomics and other aspects of personalized medicine to define destination clinics, services for international patients, and a variety of other things. It's an entire building that we expect to devote to that. There's a fourth building, which will be our technology tower. For those of you in the room, we can't do medicine without data. There's no question about that. And more data is being collected. So the real question is, what can we do intelligently to make use of that data for both patient care as well as understanding trends in an acute care facility or, in fact, our entire health system. So we expect to take most of a single tower and devote that to bioinformatics, data analysis, those kinds of efforts. A fourth area is research, and this is an R&D panel, and I'm here to tell you that Inova is in the process of expanding our research efforts fairly dramatically. What we had been a good history of research, we really want to expand that because it is our belief that that future of actual clinical care is based upon translational medicine. In other words, bringing these discoveries that my distinguished colleagues on the panel have described to the bedside, and the translational medicine is based upon starting out with basic research. So it's our belief that as a healthcare system, we have to be in that business. This is another representation of the buildings there. The fourth area I didn't describe is a wellness center. We're interested in fitness that's in a hospital context, and so we're going to be developing and experimenting with a couple of areas there. This is the description of the lab research. I described one building that we want to have as our research building that will be about 30% of our own research efforts, ITMI, the Translational Medicine Institute, but we want to work very hard to bring in colleagues from Virginia Tech, UVA, other academic research institutions to work with us on the site. We also are actively recruiting top flight commercial companies to work with us on the site. And the notion is that some of these discoveries, as our colleagues have pointed out, can become successful companies. So about 10% of the space would be devoted to entrepreneurship. whoops, go back here a little bit. Well, anyway, I thought that's the point of the story, however, is that we are going to, in the process of making a fairly significant investment. The campus cost alone was $180 million, and sadly, that's only the tip of the iceberg. But what we do need to do is expand that research effort, and so as a consequence, we are actively looking for partners in Virginia and across the nation, and I thank you very much.
So in order for us to conclude on time, we're going to move straight into the lightning round. So we'll get started with that. So if each of you all could answer, what is the single biggest impediment to innovation in bioscience? Want to start, John?
Well, I'll start that off. We need more proof of concept funds. We need what I'd call seed funding in the valley of, what we call the valley of death. Funds that can get us from the basic science discovery into the proof of concept. And I think more funding in that area will accelerate the whole system.
So my view is once a product is on the market, we need to have better alignment between the innovators and the insurance companies for paying for clinical benefit and for cost savings. And so better alignment around how we are on the same page on that.
Thank you.
Yeah, I tend to be more on the basic research side of things. So, over the last decade, we've seen a collapse in funding for basic research. And, we live in a competitive world. The People's Republic of China, for example, are set to become the world's largest spender on research and development by 2021. And I think if we're going to compete for the industries of the 21st century, we have got to invest in basic research.
Well, I certainly agree with my colleagues up here, but a slightly different point of view, which you might discern from my talk. I'm not so sure that we really have enough concentration of forces. We've got some great research institutions and great medical systems, but spread out across the state. And so the notion of being able to have a center such as Cambridge and Boston, Cambridge, or a variety of other research centers, I think is something that Virginia has lacked. And I do think we would do a lot better if we could put some of the great scientists right next to each other to be able to accelerate some of our discovery and commercialization as well.
Thank you. So thinking about the positive side, what do you see on the horizon that is an exciting development that could really impact the biosciences?
So everybody knows the first draft of the human genome was published about 2001. And if you go into the genome now, you can pull up your computers and open up GenBank. About a third of the genes are unknown. And what that means is we know the sequence, but we don't know the details about when and where they're turned on, what they're doing, who they're partnering with. So there's a huge opportunity for new drug targets in the unannotated part of the genome, and in particular, in the ones that encode membrane proteins.
My view is that, and you heard the theme this morning in some of the talks, the empowerment of the patient, the consumer, in their own healthcare, I think is a very, very positive trend, and I think that's really going to contribute to us solving many of the challenges that we have in healthcare. So I think that's very, very positive.
That's a great point.
I think the most transformational thing that's happened in biosciences in the last few years is the advent of what's called genome editing. It was discovered by two women scientists, Edna Doudna and Emmanuelle Carpentier, one in Hawaii, one in Paris. And literally, this technology allows scientists to go in and treat the genome like a word processor, editing genes, changing things. And I think it's going to be absolutely transformational to the human species over the next 50 years. And again, it was out of basic research. They found the enzyme systems for this in cave-dwelling bacteria. So you just never know where the next transformational technology is going to come from.
Well, obviously from the three previous comments, genetics and genomics are really crucial in terms of transforming healthcare and being able to understand that, but there is so much more that we need. But one of the bigger issues is that understanding which particular biomarkers and which particular issues actually are related to disease and then being able to do something about it. There's an uncomfortable number of variants that we know about that we can't do anything about it. That's one issue. The other is actually making sure that our doctors, many of them really brilliant, but also many of them were educated a few years ago, are really aware of what is out there and have the tools to be able to use some of this genetic data in actual patient care.
Great point. So just to conclude, the last question I'd like to ask, and of course the Virginia Chamber does a great job advocating for Virginia with the Virginia legislature. So what would you tell the members of the legislature in terms of things they could do to make Virginia a better community for bioscience?
Well, I think we need funds to retain talent in the state. And a lot of UVA researchers have gone other places. Our Nobel laureate, Barry Marshall, for example, was not able to be retained. And we need to keep the talent at home. And we need funds so that there's a pool so that at a critical stage when a university is struggling to retain somebody, we got a way to do it.
Capital is critically important. The R&D tax credit in this state has been fantastic. It's been very supported. The Chamber supported it. The legislature has continued to support it. Those kind of things are really, really important as we're building companies. I think at a higher level, I think our corporate tax rate in the United States is challenging as we compete with companies around the world. I think that's something we need to tackle a longer term. But I think in the Commonwealth, I think there's been great support of small companies, and I just want that to continue.
I would echo a comment John made earlier about funding for transition of technologies out of universities. Often there's, you need monies for proof of concept. You might just need $50,000 to, do a small trial on animals, but it's just very, very hard to get your hands on this type of money. So I think funding for these kinds of translational projects would be very handy.
I think we have a danger of complacency because we do have some great research universities and some great companies. The complacency, when you look at that, you say, well, we're doing okay. Truth of the matter is, if you look at the statistics across the U.S., we're not doing okay. Virginia is really, really doing very poorly when it comes to funding for biotechnology from the state level. Ohio, $2 billion. Georgia, $1 billion. I mean, the total state funding is just not there. I think that everybody realizes that Virginia is a great place and we're a great place to do business and we have great health care. But if we're going to compete on a national and international scale, we need to get serious about this and put up some real substantial money. Now, that's not easy. It's easy for us up here to ask for the money, but it's not so easy for the legislators to come up with those sources. But if we don't, we run the risk of falling behind many states who have put in lots of money.
Thank you. Well, this has been a great discussion. I personally have learned so much from working with each of you, and thank you for sharing your stories with the audience today. And I'd like to just give a great round of applause to you all. Thank you. And we'll wrap it up.
I'd like to thank